you so much, Odetta. We are shutting it down today with some soils excitement. My name is Rebecca Lybrand. I'm a faculty member in the Department of Land, Air, and Water Resources at UC Davis. And I'm thrilled to be here today to talk to you about cross-scale analyses of soil, fungi, and carbon, thinking about this as being delivered by advanced analytical capabilities. I want to start my presentation today with acknowledgements. First, can my team raise their hands in the back? Maddie, Cassie, Solvig, and Dragos have been amazing and have really worked hard to be here sharing their findings with you in their posters and presentations. I'm very grateful to them. And then overall, what better way to acknowledge folks than with photos? And the mesh bag project that I'm going to be mentioning today and talking about involved deploying hundreds and hundreds of mesh bags across multiple states, ranging from Sonoran Desert, where our mesh bag deployment day went into the evening. So we had a nice evening hike out from our desert scrub site right here, to figuring out how to deploy mesh bags in Southeast Alaska when we're worried about bears digging them up, from black bears to even brown bears, and how to navigate this using float planes to reach some of the sites that we actually deployed some of these samples at, all the way to the Southeast in South Carolina and Georgia, where you would think I intentionally booked every deployment and retrieval day when there was going to be weather, because every time we've been there, it's been a complete downpour. And I'm just so fortunate for everybody here on the screen and everyone I listed on that slide before for making this work possible. This was probably my favorite slide that I created in preparation for this talk, because it gave me an opportunity to think about all of the wonderful collaborations and conversations and discussions and proposals and publications that have come out of working here at EMSL. And this has transformed my career. And I find it so inspiring as a soil scientist to be able to come here, to bring students, to bring the team, and to work collaboratively with all kinds of different scientists here. So I'm just very grateful for the opportunity to be at EMSL once again. And for all of the folks you see mentioned here, I couldn't even fit everybody into this slide. So there's several people who were listed on my first acknowledgement slide as well. So again, just over the moon to be back, and we're really excited to get going on our next user proposal. So we're soil scientists. We like to go out, we dig soil pits, we do soil profile descriptions, we think about soil landscape relationships and the importance of soil here on Earth. Some of our work has even gotten into thinking about ground nesting bees and how soils provide that habitat to support different types of ground nesting bee populations. In addition to that, our work has been pushing more and more towards the importance of microscale to nanoscale processes and understanding those as controls on mineral weathering, nutrient cycling, or even carbon distribution. And I don't have time to talk about it today, but I would love if someone in the audience asked about our work that we're doing to bridge soils and the planetary and space sciences. I may have a couple of slides at the end after the question slide that goes into this if anyone is interested. But overall, one of the key components of this talk and the structure of this presentation is to highlight the importance and the critical need to study soil through university government partnerships. And you will see that I've emphasized here with an emphasis on the strength of EMSL's capabilities and expertise for better understanding microscale processes. So one of the key questions, you can picture this as like the umbrella question driving a lot of work within our team, is thinking about how do abiotic and biotic environments interact to initiate mineral weathering and the formation of soil in Earth's critical zone. And we have been thinking about these questions and looking at soil development across a range of different climates. This work started with an environmental gradient just outside of Tucson, ranging from Sonoran Desert up to subhuman mixed conifer forest. It's expanded to the Southeast, where we are looking at soil formation and development in Georgia and South Carolina. And we have focused a lot of attention in recent years in Alaska, spanning coastal temperate rainforest, and then a discontinuous to continuous permafrost gradient through a study led by former student Dr. Aaron Rooney. And then finally, through a current collaboration with NASA, we're also looking at soil formation and microscale weathering features at Mars analog sites across an Icelandic environmental gradient, focused specifically on cold deserts. So today, we're going to be focusing on work that was conducted in Arizona along this climate gradient, specifically the mixed conifer and desert scrub sites, as well as the Calhoun Critical Zone Observatory in the Southeast, so specifically humid hardwood forests in South Carolina. And then we'll also be making a comparison with coastal temperate rainforests in Alaska. And just to set the stage for that, this is in Southeast Alaska. The permafrost work that I mentioned, we won't be highlighting that today, but we do have some papers demonstrating soils work with EMSL capabilities between a discontinuous and continuous permafrost gradient as well. But when you hear me mentioning sites, it's going to be Arizona, South Carolina, as well as Southeast Alaska and the coastal rainforest. So one of the primary processes driving soil formation is mineral weathering. And as soil scientists, we are really excited thinking about all of the different mechanisms driving how you can go from having calcium, phosphorus, magnesium, and other types of elements bound up in rock, undergoing weathering processes, and becoming available in the soil for plant uptake to support life. Weathering is important. And given this has been summarized in Elements in a 2019 article where they were emphasizing how this is an interdisciplinary type of study. And this is actually highlighting all kinds of different disciplines here with pedology, or the study of soils across natural landscapes, here in the black box. And I am pushing to say that soil scientists and pedologists can actually go down further when we're thinking about spatial scales, especially into the nano to micro scale side of things. But we need university government partnerships to really be successful with this, because we're in a full room here of experts on these different types of analytical techniques. The other thing that I wanted to briefly mention, I could have an entire conversation here just on weathering and all of the different types of chemical weathering and physical weathering and freeze thaw and all of these different aspects. But there were a few key terms that I'm going to be using when I present the results today that I want to make sure that we're all on the same page with. So this is a diagram of different types of fungal weathering mechanisms. And I wanted to bring your attention especially to this term here, pressure dissolution, or thinking about biomechanical weathering. So this is the physical force that fungal hyphae apply when they're growing through the surface layers of minerals, growing along the edges, as you can see here, and really growing between these different layers and physically pushing some of these layers or disrupting the mineral status of some of these different types of minerals that we've been studying. The other type of dissolution that I wanted to highlight is biochemical weathering, which would be the direct fungal grain contacts when organic acids or types of exudates are being released, or more indirect weathering where fungi are releasing these types of organic acids more indirectly to drive weathering processes in the soil. And then I also want to emphasize that fungi can also produce biominerals. And so this is documented as an example here as oxalate, as well as different types of coatings or EPS that you see represented with the light gray here, or exopolymeric substances. So these are several different observations and features that you may see in some of our results today, especially that biomechanical weathering component. So we've talked about the importance of mineral weathering across spatial scales, and we've thought about fungal weathering specifically. And I just wanted to briefly highlight the importance of mineral weathering across agencies. So when we think about NSF, we are talking about biological weathering, physical weathering, different types of weathering processes and how to distinguish these. NASA is also interested in mineral weathering. It's just a different type of terminology. In the astrobiology strategy, especially, NASA is emphasizing the importance of studying the impact that fungi and bacteria and other types of microbes have on minerals. The signatures that are left behind by microbes on minerals is something that NASA is really interested in. At the same time, NASA also wants to identify false positive biosignatures. They don't want to go to Mars and say, we found evidence for life, and then find out it was actually formed from a water rock weathering reaction. So at the same time, NASA is funding all kinds of studies in Mars analog sites here on Earth. And this is actually one of the studies that we're involved with in Iceland. And last but not least, but the Department of Energy is also interested in mineral weathering. If we think about it in terms of biogeochemical processes that are occurring in the soil, and then also this push for a mechanistic understanding of below ground processes, again, specifically emphasizing soil processes and the importance of mineral microbe interfaces. So the point today of today's presentation is to think about how we disentangle the abiotic and biotic processes that transform minerals and release life-supporting nutrients to the soil. And we're specifically focusing on mineral water microbe interactions. So before I jump into what we did with EMSL, I want to talk a little bit about what we accomplished as soil scientists across some of these different environmental gradients. We set up this environmental gradient study outside of Tucson, where we had desert scrub environments, and we were taking soil descriptions from here up to oak woodlands, all the way up to this mixed conifer system. We collected samples, and we were looking at the mineralogy, we were looking at the clay mineral distribution, and we were also looking for evidence of elemental loss compared to the relative parent rocks that we sampled. And overall, we saw a greater degree of elemental loss, as well as clay mineral production occurring in the wetter, more productive mixed conifer sites, which we would expect. In the mixed conifer sites, we also saw that the downslope positions that were wetter and received more soil and water distribution across the landscape were also more highly weathered. So these were observations that we were linking back as a chemical weathering process. At the same time, we were also looking at carbon. We were thinking about the distribution of carbon, the persistence of carbon, and the partitioning of carbon across different types of carbon density separates. And then finally, we did perform a microscale study comparing soil sampled from the desert scrub environment up to the mixed conifer site. And this is one of my favorite photographs or images that came from that work is this biotite grain right here. We captured this with the electron microprobe. And this is a perfect example of frayed edge weathering. And since we analyzed it with a microprobe, we could actually detect the loss of potassium from the inner layer space at the edges of this particular grain. So even in the desert scrub site, we did see evidence for mineral weathering at the microscale. But overall, we saw a greater proportion of mineral alteration occurring in the higher elevation sites, which we attributed to water and the productive system. However, that is basically where we had to draw the line. We didn't know what exactly transformed these grains. Was it all water rock interactions? Was there fungi also engaged in this type of weathering? These are the types of questions that emerged from these different papers that we worked on. We also extended this work to perform some field and soil morphological assessments through a collaboration with the Forest Service. And this was located up in coastal temperate rainforest near Juneau, Alaska. And the Forest Service was specifically interested in understanding upland soils and how the soils form and distribute carbon, even if they're formed on different types of lithologies. And so this is a metavolcanic rock and a slate rock. So we were comparing a few different lithologies and what the soils look like in the soil profile form. So one of the students working on this project dug with the team 18 soil profiles in really challenging, difficult to access terrain. In those images, the slopes are from 30 to 80% upland slope. So this was quite a character building type of experiences to be doing field work in. At the same time, we were seeing consistency across all of the different lithologies with respect to the soils. And so even though the soils differed, differences in slope, and we were also comparing north and south facing aspects, for the most part, the outputs were similarities in soil development with a patchy E horizon or leach alluvial horizon, and then some subsurface some subsurface carbon accumulation in these mineral soils. The other point here is across the board, regardless of lithology, we saw relative consistency in mineral soil carbon accumulation. And the amount of carbon that we saw in the mineral soils was also actually greater than what we saw in the organic O horizons. So as the soil scientist side, our main takeaway here was that the climate, the intense wet type of climate regime in Southeast Alaska, combined with a productive forested system, was driving this consistency in soil formation, as well as consistency in the distribution of the carbon. We actually had another student jump into the project, Julia Jansen, who extracted carbon fractions. Would there be a way to turn off? I don't know why we have the automatic narrations going. All of that was unchecked. We good? You kept getting previews before I got to my takeaway points. So we had to get back into the pace of that. Okay, so overall, as the soil science, that survey wrapped up, we were saying bioclimate is swamping out any signature that we had from the lithology. And then we had an additional student jump in and extract carbon fractions from these different slate and metavolcanic soils. And she actually started seeing, when comparing the freelite, the occluded, and the mineral fractions, differences in partitioning of carbon. And even when we've just started getting our radiocarbon analyses back, we may even see some differences in the persistence of carbon in these particular samples. And this is where one of our EMSL collaborations comes in, because we were actually able to send some of our carbon fraction samples up to EMSL, and Odetta was looking at these using the scanning electron microscope. And as we started thinking about why would we see differences in carbon persistence or the distribution of carbon, especially in our mineral samples, we were comparing the metavolcanic mineral grains to the slate. And you can already see the differences in complexity of these two different types of soil minerals. And here there's a lot of pitting. There's a lot of complexity here in the metavolcanic grains compared to very smooth surfaces in the slate samples. Now, we still saw organics. We still saw a lot of carbon in these samples, but there was a much greater degree of complexity and differences in the overall morphology of the grains that we took a look at. The other set of micrographs that had us so excited from the soil side was actually taking a closer look at our free light fraction carbon fractions. And it was completely covered in fungi. There was fungal activity all over these different types of materials that we were looking at. And then we were also looking at the occluded fractions. So these are samples that were sonicated, and we still have that issue. If you hit review and just make sure none of those boxes are checked on the PowerPoint. So for our occluded fraction, this is a material that's undergone density separation. And then at the same time, these materials were also sonicated. But even then, we were able to see aggregates or micro aggregates of organic matter, and again, fungi distributed throughout these types of samples. So overall, between our work in Arizona, where the concluding points were, okay, what are these microscale drivers of mineral weathering or soil formation? Is it more on the abiotic side, or is it more on the biotic side? And then at the conclusion of some of our Alaska work, we also had the same types of questions related to soil formation and the role that specific types of biological agents, such as fungi, may be playing in the system. And that is what led to us producing these mesh bags and putting them out across all kinds of different ecosystems and environments for study, for incipient weathering types of analyses. And we put out nylon mesh, and we initially tried a few different sizes of nylon mesh, from coarse 35 micron, where we planned that this would allow in fungal hyphae, but exclude plant roots, all the way down to fine mesh at 0.5 micron. And that idea is that microbes could move in or have water flow through. And the idea is that it would exclude fungal hyphae. But some of the most beautiful images that we got of fungal grain interactions actually came from our fine mesh bags. So there are definitely some infiltrators that moved right across the 0.5 micron mesh size. So with that said, though, we did deploy all of these mesh bags in the surface mineral soils between 0 and 10 centimeters across all sites. All of the soils that we looked at were derived from granite. So these were granitic soils. And in terms of the different types of rock materials, we had basalt that we put out. We viewed this as a bait. It was a fine grain material. The microbes are interacting in the soil likely wouldn't have seen it before. Fine grain, lots of basaltic glass, and potential sources of calcium, phosphorus, iron, among other types of elements. We also put out a quartz sand. This is coarse grain material, and we were viewing the quartz sand as a background control. We were able to see what was moving in and out of the samples, what was coating the samples, and it was also a space for documenting any kind of fungal foraging that may be going on, since there wasn't a specific type of element to be drawing them in. And then finally, we put out granite. This was a coarse-grained, basically fresh supply of bedrock material at the soil surface. And this was including sources of potassium, magnesium, iron, calcium, among other types of elements. Within each field site, we deployed these in upslope and downslope landscape positions, and we actually repeated this upslope, downslope pairing 3 times within each ecosystem. And the red squares represent 24 mesh bags each that were retrieved at different time points. And overall, our timeline is that we retrieve bags after 1 year, after 3 years, and then most recently, we retrieved a series of mesh bags that were out for 6 years. So this was a really exciting moment when we got to go back and get samples out of the ground that had been there incubating for 6 years. And as a quick reminder, these are the 2 sample areas that you'll be hearing me reference: the desert scrub and mixed conifer in the Catalina Mountains, and then the Calhoun Forest in South Carolina. And we were interested in these because we were able to set up similarities in the distribution across these north-facing hill slopes with respect to upslope and downslope comparisons, and then also that climate gradient, mean annual precipitation of 25 centimeters in the desert, up to 85 centimeters in the subhumid forest, and then 127 centimeters over in the humid hardwood forest in the Calhoun in South Carolina. All right, this is one of my favorite sets of microscopy images to share. So picture producing hundreds of these mesh bags, flying all over the country, spending hours deploying them, flying back a year later, retrieving them. And during that same time frame, there's been challenges. You know, reviewers are not quite on board, thinking there's going to be no fungi in these bags. There's not going to be any evidence of any kind of grain interactions, no biological activity. But EMSL believed in us, and we were able to get a rapid proposal funded to bring our samples here. And so we literally went and retrieved bags in South Carolina, and I flew almost directly to EMSL. And I was able to set up time with Shuda on the helium ion microscope. And these are the first images of fungi that we detected in a basaltic mesh bag. And look at that level of detail. We were so excited about these outputs and some of the different observations that we were able to make with this particular instrument. So one of our first outputs from this work is that helium ion microscopy is a promising tool for probing biotic mineral interfaces. And this is just a quick look at what this looks like. And then this is an example of some of the scales that we were crossing using the helium ion microscope with all of this fungi distributed across different grains, and then honing in on one specific interface all the way down to the nanoscale of study here. The other thing that was really helpful with using the helium ion microscope is the contrast, the level of depth that you're able to capture with these images, as well as the fact that biological specimens are not as damaged using this technique compared to other types of microscopy. One of our biggest points that we've emphasized in our 2019 paper is that we actually detected evidence for biomechanical weathering. So that's that term I used at the beginning. That's kind of the physical weathering of fungi growing along grain edges or grain surfaces. We detected evidence for biomechanical weathering in all of our sites. After only 1 year of deployment in the field, we were seeing evidence for biomechanical weathering in the desert scrub, mixed conifer, and the humid forest sites. And it presented itself in a few different ways. You can see the fungal growth here and growing between the surface mineral layer. You can also see evidence for fungi growing along the edges of grains, utilizing probably a pre-existing crack in the mineral material that was put out. And then also, this is another example of biomechanical weathering right along the edges of this grain that we were taking a closer look at. So overall, we were able to detect evidence for weathering after only 1 year. And we continue to see this type of weathering in addition to other forms as we went on in our mesh bag deployment study. And then I also wanted to highlight that Cassie Collins is a graduate student with our team who is starting to take a look at developing a fungal absence presence study and being able to track down fungal activity to specific mineral types. And we're actually going to be working on this through our upcoming user proposal. So stay tuned for more information on that. So the next step, once we were able to start using the helium ion microscope, we wanted to know more about the minerals. We wanted to know more about the elemental distribution of what was happening in the particular samples that we were looking at. And so we were able to use the helium ion microscope to capture images of the morphology, the details. Certain types of features were catching our attention here, such as this aggregated material that appeared to be adhered to the fungal structure. But we weren't able to get any information on the elemental distribution using just the helium ion microscope. So we went on to create this co-location reference grid locator technique that allowed us to take the same exact interface on the helium ion microscope and go locate it again on the scanning electron microscope to detail the elemental distribution through mapping as well as point analyses. So we were able to combine that amazing, beautiful imagery with the helium ion microscope, with the SEM, to understand that this fungi was basically associated with a biotype micrograin. And this adhered material actually was not just sloughed off organic material. It appeared to be aggregated iron with some particles of titanium. So we went on to apply this technique and to extend it even more. And this was an interface that we were especially excited about because we were seeing not just the fungal growth across a grain, we were seeing these different types of minerals on both sides of the fungi on these samples, and they were iron-rich minerals. So our initial hypothesis is that we may have been seeing evidence for biomineral formation. And then through a collaboration with the transmission electron microscopy lab and Lybrand specifically, we were able to open up a vertical cross-section of that same interface here to look at the fungi. And much to our surprise, these grains that we thought were just on the surface were actually embedded in the volcanic glass matrix. And further analyses identified these as titanomagnetite inclusions. So these were actually minerals that were formed with the basaltic rock. But there were several points on these different grains where the fungi were specifically interacting with these locations on the volcanic glass where we had the magnetite inclusions present. And our hypothesis is that the presence of magnetite inclusions is known to weaken the structure of volcanic glass. And so we were thinking that the fungi are actually utilizing that area to mine out nutrients or elements from the volcanic glass because it's more susceptible to weathering with the inclusions being there. So that was one of the main points from that work. And this is what that interface looked like in some more detail. This is where the fungal hyphae was interacting with the grain. And here are some of the outputs. We were able to detect different forms of the titanomagnetite. So we see dendritic as well as subhedral. And then I also wanted to bring your attention to this dip here in the surface and that that darker gray layer here. We were actually looking at this as a form of biomechanical weathering. That dip or the morphology at the grain surface, we think that was resulting from the fungi releasing organic acids and weathering down into the grain surface. We also had the opportunity to work with Danny with the Atomic Probe Tomography Lab to check to see if there was any kind of dissolution between the magnetite in green and the volcanic glass in gray. And while we didn't see any evidence for dissolution or weathering, these are very abrupt boundaries here, we did see evidence for the abiotic enrichment of phosphorus, but mostly calcium right along this type of interface. And this likely resulted during the formation of the rock material itself. But these are what some of the outputs look like, comparing volcanic glass over to the magnetite, and these were the boundaries between those particular samples. And we also ran a second grain just for additional data and analysis, and we saw something very similar. This particular sample, the emphasis again was on the abiotic enrichment of calcium. So overall, we had a few main takeaway points from this work that we were able to accomplish by combining scanning electron microscopy, the focus ion beam technique, as well as the transmission electron microscopy and the APT technique. First was we observed the abiotic enrichment of calcium and to a much lesser degree phosphorus at the glass magnetite interface. We also saw evidence for biotic aggregation. Those exudates coming off the fungi, they're sticky. You could see all kinds of mineral particles and other types of materials sticking to it, and we really do think this is driving some incipient aggregation of these grains that were put out. And then we have magnetite being exposed on these grain surfaces that could be enhancing fungal mining or even exposure to redox reactions. And then finally, we were thinking about this in terms of alteration by fungi, the fungal mediated hydrolysis along these basaltic glass magnetite surfaces. So our ongoing work is continuing to look at fungal driven weathering in collaboration with both the Forest Service as well as an NSF career project. These sites have been expanded from Juneau to Prince of Wales Island in Alaska down to Calvert Island, British Columbia, and also continuing work at the Arizona and South Carolina landscapes. We're also so fortunate to have Dragos on board as a professional scientist with the lab who has expertise in preparing these types of samples, because we just put out a whole suite of mineral chips that were pre-characterized and imaged before deployment. So we're going to be able to look at both ground rock as well as these cleaved minerals in a pre-post deployment type of comparison. And then we've got Maddie on board, who's going to be looking at these mesh bags using XCT to identify any evidence for incipient soil formation. And then Cassie is on the team thinking about high resolution microscopy to really get into some of the selective bioweathering that we may be observing with the fungi between these different rock types. So to summarize, soil matters, and we've been working a lot in this realm and thinking about different types of soil, but moving more and more into this space. And so in summary, today we talked about abiotic and biotic weathering with an emphasis on fungal driven weathering and being able to develop techniques and capabilities and collaborations with EMSL specifically to be able to co-locate and better understand these fungal grain interfaces, especially how heterogeneous some of these processes are. So as soil scientists, we're going to keep pushing the boundaries of this box here. I'm especially thinking about this more at the micro scale, but we couldn't do it without amazing collaborations like what we have here at EMSL. And so with that, I'm happy to answer any questions. And then also, if you are interested, the Critical Zone team made a comic about the mesh bag project, and I pushed, and they even included PNNL and specifically EMSL. So you can see Odetta here featured with the scanning electron microscope. So you can look up that comic if you want to read more about this project in a comic book form. But thank you for your time and for hanging on through the end of today.
Turn now. Thank you, Rebecca. This is fantastic presentation as always. And I am looking if there's any questions about the work that was presented here. Well, I will start if there is nobody. Yeah, others get ready to ask question. But you mentioned that you also are interested planetary connection between what's happening on Earth versus potentially what is happening in other. I'm curious if that is a work that you are bringing with the user proposal here in EMSL that is coming up in the next year. I mean, this year, actually, 2020.
Yeah, we actually want to apply to it for an additional proposal, especially once we have some of our Iceland samples back. So going through all the permitting and that type of thing. But we do plan to bring samples here if we can get a user proposal funded, focus specifically on that.
Yeah. And would there be, again, like the mesh bags that have been buried in there? Okay, so that would be
yeah. So we actually have a set of mesh bags currently deployed in Iceland in a site. It's about 2 million year old volcanic flow, and then one in a much wetter section of the cold desert climate. So it would set up a really nice basalt weathering type of comparison.
Yeah, thank you. Aaron?
Beautiful talk. Those are amazing images. I can't help but look at all those fungi and think, who are they? Who's doing this work? I'm just wondering, I'm sure you thought about that too. Have you guys looked into that? Or
we have dreamed of this, but we don't have the expertise on the team. So this is something that I even have bags that we've preserved at -80 and just go, who are the communities? And yeah, we would love to do this type of work, but we need to add the team members who have the expertise, and we also need to line up the funding for that to support students or postdocs or somebody to work on it. But yes, that is definitely something that we're interested in, and I think it would add so much from the biological side of things. Completely agree.
Yeah, and I don't know if James is here, but he talked about 1,000 fungal proposals. So you might just.
I was thinking about that as he was presenting. Actually, he was like, "Oh, I wonder if we could bring some of those mesh bags over." Thank you. Perfect. Yeah.
The organic acids that you alluded to, do you know what they are?
Nope.
Great.
Yep, that's something else. We've been talking some to Sharon Billings and thinking about how we could measure those as well. So again, we're soil scientists, specifically mineralogy, but we would love to have more of a biological component, and especially looking at who's there as well as the types of organic acids. Yep.
I want to thank you again for a fantastic presentation, and I will pass